The overall goal of this protocol is to establish primary mixed glial cultures from adult mouse spinal cords for in vitro studies.
This method provides us an in vitro system to investigate the roles of glial cells in neurological diseases that involve pathological changes within the spinal cord, such as neuropathic pain and multiple sclerosis. The main advantage of this technique is that the glial cultures are prepared from adult mouse spinal cord, providing a system that is more accurately reflecting the in vivo conditions. Demonstrating the procedure will be Jennifer Malon, a technician from my laboratory.
Working in a tissue culture hood, transfer 4 mouse spinal cords into a petri dish of HBSS. Use sterile scissors and forceps to cut each of the spinal cords into small pieces. Then, transfer the pieces to a 50 ml conical tube containing papain DNase enzyme mixture. Avoid transferring HBSS to the enzyme mixture, as this may result in decreased performance of the enzyme.
It is crucial that tissue pieces are well digested to obtain a single-cell suspension. However, overdigestion will result in fewer viable cells. Each lab needs to perform pilot tests to determine the exact digestion time based on what works best for their cells.
Next, gently vortex the tube and then incubate at 37 degrees Celsius for 1 hour with orbital shaking at 150 rotations per minute. Following the